Greetings everyone, my name is Shoaib Rai and I am the biology faculty from Nucleon Coaching Institute. Earlier we finished on 4 topics that were reproduction in flowering plants along with this entirety, that is the development of the embryo and the double fertilization event, which is important for your board's examination. Then we came on to our ecological pyramids, the differences between the pyramid of energy, biomass and number. Moved on from that to recombinant DNA technology. How do we basically introduce a foreign gene into our preferred one to create more number of copies. Okay. Then we moved on to Hershey and Chase experiment, which is very important from your molecular biology. Okay. Now, with that itself, I have brought you some more 4 topics that are very important for your boards as well as for your NEET, and as such, we will be going through and discussing them along the way. Okay. The first thing that we would like to, or I would like to discuss with you is that the first in our second part is the menstrual cycle. Okay. Now, in simple definitions, what is menstruation and what is menstrual cycle, we all know. Okay, and some of the ladies that are watching this, they already experience it. Okay, so in a proper term, how do we write it? Okay, menstruation is a process. Okay, is an event that involves the rupturing of endometrial lining, the ovulation, the follicular part and the post-ovulation period, okay, that follows. Okay, so we will be discussing this, and in my suggestion, it is best explained from your books itself, from this diagram that I have taken. Okay, now this diagram, the best thing about this diagram is that it properly exhibits all of the stages that are important for you to consider, that is the hormonal levels from both your pituitary and your ovarian hormones. Okay, ovarian hormones are simply those hormones which control the activity of the ovum, okay, or the development of the ovum, along with the pituitary hormones which help the follicle to develop. There is a difference between the ovum itself and the follicle itself. That is, follicle can be understood as the cover, okay, or as the part or the sac that is present in which the ovum itself is developing, okay. And the ovarian hormones also control the uterine muscles, okay, the uterine wall layers, okay, everything, okay. Now, if you see from this part itself, okay, that in the first few days of the month, okay, you will find that in pituitary hormones, FSH stands for follicle stimulating hormone and LH stands for luteinizing hormone. The concentration of FSH is much more higher as compared to your LH in the first few days. Why? Because you need this to stimulate the growth of the follicles from this stage to develop into a mature follicle where it is filled with graphene fluid. Okay. Ovarian hormones we will simply see as estrogen and progesterone. In this case, estrogen is much more higher in concentration as compared to progesterone, but they both steadily increase. Okay. Moving on to the follicular phase or the proliferative phase. Why do we name this proliferative phase? Because in this stage itself, it proliferates or moves, okay, moves into the next mature part where it needs to rupture, where the follicle needs to rupture in order to release the ovum. Okay. So this is controlled by the peak rise that you see or is visualized in the 14th day, that is the end of your follicular phase, okay, is marked by the steep rise of pituitary hormones such as your LH and FSH. Now, there is a difference in the quantities of the rise, that is LH rises much more steeper as compared to your FSH, whilst that of estrogen and progesterone rises in much much more higher compared for estrogen, whereas progesterone simply does not rise on the 14th day. Okay. Why? Because estrogen, luteinizing hormone and FSH, those three hormones need to be at their peak or need to show a steep rise in order for the ovulation period to begin. That is our next phase, that is the luteal or secretory phase to begin. Okay. Now in that part itself, if you see in the uterine muscles, okay, for the first few days you will see that the menses, that is the rupturing of the blood vessels in the uterine muscles, that is the endometrial lining, ruptures in order to release the blood. Okay. After that, in this period itself, okay, it rebuilds itself, it regains itself as the follicular phase develops. Why? Because the ovulation marks or expects the body to be ready for the upcoming pregnancy, okay, or the upcoming fertilization and the implantation followed. If there is no implantation, it ruptures again, releasing the blood in the next one and the next cycle. Okay. In this case, you will see that after the ovulation has started, okay, there is a steep decline. There is a steep decline of the percentages of the part, releasing into, and when the ovum starts to move from the fimbriae to the uterus through the lining up, and in that stage itself the hormonal levels start to drop on all those hormones that were at the steep in the ovulation period. Okay. Now what happens is the same situation is now reversed, wherein progesterone starts to increase only and only if there is no pregnancy. That is, if there has been no fertilization and implantation. To be very precise, there has been no implantation. That is marked by the increased concentration of your progesterone over estrogen. Okay, over estrogen, and then the next cycle continues. Okay. Now this itself is very important in the sense that it can be asked from you directly in a 5 marks question to explain the menstrual cycle along with the levels of hormone and their effects on the uterine muscles. So I would expect that all of you go through with it. If you don't understand anything, please let us know in the comment section and let us move forward with the next step. Our next topic. Okay. Now this is lagoperon. Okay. Now the first thing that anyone or a basic question arises is, what do you understand by an operon? Okay. Operon are simply those genes for prokaryotes. Why prokaryotes? Because these are the house controlling genes or household genes. Okay. Now, if I tell you household genes, what do you understand by household genes? It simply means that they do not switch off, okay, or they do not stop functioning, okay. They continue to function and are required for the regulation of every biological process, okay. And one more thing that is different from a prokaryote and a eukaryote is in their cistronic nature. Those are polycistronic and we are monocistronic, okay. Now, in this case itself, the lagoperon is a inducible operon. That simply means that there is an inducer in whose concentration, if the increment is visualized, increment in the concentration is visualized, there is a steep increase in the concentration of the inducer, it will trigger the operon to function. Why? Because there is already a repressor. The function of a repressor is to stop the activity of the promoter region, because this is a gene, right? And gene has a promoter region. Okay? This stops the activity of the promoter region, repressor. Only when the inducer starts to build up, the concentration of the inducer starts to build up, and in this case, lac is the inducer. This lactose is the inducer. Okay. Repressor mRNA will produce repressor repressor which binds it as it is written, okay, preventing the transcription of the operon. This is overcome by the inducer, in which case it will be our lactose, okay, which basically binds to the repressor, pulling it away from this site, that is your promoter site, okay. And now the transcription can begin. Z, Y and A, these have specific functions. Z is responsible for beta galactosidase, Y is responsible for permease, and A is responsible for transacetylase. I will tell you what are the functions of this. Galactosidase, okay? Galactosidase is a type of an enzyme that is responsible for breaking your lactose into further simpler compounds, okay? Is ACE, okay? Remember, if you don't, that ACE types of compounds, they are mostly responsible for ubiquination. What is this ubiquination? It is simply the breaking of those heavier molecules into smaller parts or into their smaller fragments, simpler fragments, okay? So beta galactosidase basically performs that, okay? It basically converts that into glucose, lactose into glucose. Why? Because this is also required for the process to stop. We'll come to it. Okay? Then there is permease. Permease simply allows for the lactose to be in or taken inside. Okay, lactose to be allowed entry. Okay, inside the cell for the repressor to be taken away by the inducer itself. Okay. And transacetylase, as is the name, simply performs transacetylation, that is addition of the CH3COO group, okay, on the trans phase of the, I think this will be a bit too much information, but still try to follow. Transacetylase simply performs the acetylation on the trans phase of the repressor protein. Why? Because it needs to change the structure in order for it to continue transcription. Okay? For it to continue transcription, and this is the mRNA that is going to be produced. Okay? That is going to be produced, and this is the complete process of the lactoperon as per your syllabus in class 12. Okay? Lactoperon is also a very important chapter, okay, and a bit confusing if you don't know, or if you don't know how to or how does this function work. I would suggest that you view this video, okay, ask me doubts if you have still, okay, but the process itself is very simple. There is a repressor, and to remove that repressor you will need an inducer, okay. Now, from the word itself, inducer, it needs to entice something, okay, or incite something, okay, promote something. So, what it does is, it simply takes this repressor along with itself, okay, and makes way or makes a clear path for the next process, that is the transcription to begin. Why transcription? Because after all, this is a gene, and that is why a repressor is bound to the promoter region, not to the structural region or the terminator region. Okay. Now, moving on to the next part, that is our polymerase chain reaction. Okay. Polymerase chain reaction was given by Gary Mullis. Okay. This, keep it as your homework. Tell me in the comment section that in which year was PCR developed, okay, by Gary Mullis. Okay. Now, PCR itself is the process by which we can make thousands and thousands of copies of one single originator or one single sample. Okay, from that one single sample we can make more than 1 billion copies. Okay, depending on the amount of time you have let it run. Okay, now it works on 3 basic principles, that is your denaturation, annealing and elongation. Okay, denaturation simply is that in this we are taking double-stranded small fragments of DNA. Okay, double-stranded small fragments of DNA which needs to be broken into single-stranded fragments. Why? Because I have told you in the earlier classes itself that in whatever condition a double-stranded structure is not suitable for any kind of biological process. It is not suitable for transcription, not suitable for replication, not suitable for your translation as well. Translation although is not related to that, but since transcription is, it is still based on that. Okay. So what we need to do is we need to break its structure into what exactly? Into single stranded forms. Okay. For that we need a very high temperature. Okay. And for that simply we will need 94 to 96 degrees C, preferably 92 also. In some books you will find 92 as well. Okay. 92 to 96 degrees C is the preferred temperature for denaturation. Okay. Now after denaturation comes annealing. Okay. After denaturation comes annealing. Now in this annealing part, what do we do in this annealing part? Simply breaking it into single strands is not going to help. It is not going to help in the process of replication. Why? Because at the end of the day we are going to replicate it, right? So in this replication what is happening is that we are going to add a polymerase that has the ability to withstand such high temperatures. Why? Because if you remove that temperature, they will again bind to each other. Okay, they will again bind to each other and our purpose will be defeated. So in that maintenance of temperature and for such a polymerase that can withstand such high temperatures, we use dark polymerase. Okay, short form for Thermus aquaticus, the microbe from which this polymerase was taken from. Usually it is found in the springs, hot springs. Okay. Now Thermus aquaticus has a temperature of 72 degrees C. It can survive up to 72 degrees C, that is the thermal springs or hot springs. Okay, the temperature of the hot springs. Now from this is very stable. The dark polymerase itself is very stable. We add it in our mixture along with a 15 to 20 base pair or a 15 to 20 nucleotide long primer. Why do we give primer? Because this is the process of DNA replication. Even in the previous DNA replication process, we have learned that it requires a primer for the identification of the opposite sequence or the complementary sequence to which a 5' end is or a 3' end is provided for the dark polymerase or basically DNA polymerase to act and keep on attaching or keep on joining the complementary nucleotides. Okay, or keep on adding the complementary nucleotides. Okay. So after this has done, annealing must be done in not at this high temperature. 68 is a very high temperature. We do it in about 52 to 58 degrees C, maximum of 60. Okay, we don't go beyond that. Why? You will find it in your higher classes. Okay. Then we move on to elongation. Okay, the last step and the most crucial step. Why? Now that we have already joined them, okay, or already started creating the complementary pair, all that is left is to create more number of copies. And to create more number of copies, we take it at elongation at 72 degrees C. Okay, elongating them or simply allowing for the process to continue in cycles. Okay, in cycles, and in each cycle it simply, just suppose from 1 you have created 2, from 2 it will be going to 4, and from 4 to 16, and you get the exponential power, okay, of 2 to the power n, in which it will be created. Okay, but the thing is that we don't simply create it for 2, we create it for more than 2. Why? Because there will be at least a 15 to 20 base pair long segment that we are actually going to increase the amount. This is our desired fragment that for which we are taking the PCR. Okay. Now from the figure itself, if you see, this is our double stranded form. Original DNA which needs to be replicated. we basically divide it or basically create a single stranded form onto it. A primer is added. Okay, this blue represents the primer, and after the primer it gives you that 5' to 3'. So this basically gives you a 3' end for the polymerase to act, okay, on both the sides. And after the polymerase has acted, we simply keep on repeating this process for cycles to generate these number of maximum copies, okay, which comes in elongation. And this is the short but a very informative version of your PCR, okay. Moving on to the last part that I need to discuss with you for this video is carbon cycle. And this is also a very simple one because you all have studied this, or you have kept studying this from your class 5, from your class 4. It is simply that, and it is still the most easiest part. Ecosystem is still the most important in terms of the questions, and it is simply still the most easiest part if you know how to write the answers, okay. What is this carbon cycle? Simply the sequestration, okay. What is the proper term that we use? That is sequestration or fixation of carbon, okay. And the entire cycle of simply changing the carbon onto a more usable form and replenishing the source again, okay. So this is what is happening, right? From carbon dioxide in atmosphere, okay. Then it is again taken back, air, sea exchange, respiration also exchanges it, okay, from marine photosynthesis, okay. Oceans and ocean sediments make it to microbial respiration and decomposition. From fossil carbons also it is the same. And from leaching and runoff. What is this leaching and runoff? When rain falls on a soil which does not possess your topsoil layer, something called as humus, okay. Then a part of the nutrients or a part of the minerals are leached off. That means simply washed off, okay? And that simply is in that form. Now what are those minerals that can be washed off? Why? Because calcium or carbonatic forms, okay, calcium and carbonatic forms are very easily washed off because they don't dissolve well in water, okay. You have seen it in your experiments from class 10th also. They don't mix very well in water, okay. That is why they are simply washed off, okay. Now this soil carbon can have two ways on how it can be fixed, sequestrated, fixed, okay. It can be fixed as fossil carbon, which we still burn as fuel, fossil fuels, or it can go through microbial respiration and decomposition to be made into leaching or runoff, which then again goes back to your atmospheric carbon, okay. This is simply carbon cycle. You will find nitrogen cycle also. Nitrogen cycle is also much the same except for all this soil carbon sequestration. Nitrogen, the biggest contributor or the biggest percentage of your atmospheric air is your nitrogen, okay. And in that itself, who is the one that takes nitrogen the most? That is plants itself, okay. Plants are the source of or the reason where exactly nitrogen is sequestrated or fixed more. But there is another organism or a class of organism that does it even better, that is your microbes. The reason for all these elements or reason for all these elements to be fixed is your microbes or are your microbes. Soil is also fixed by microbes, okay, and so is your nitrogen, okay. So these are simple questions, okay, that can be asked from various topics, okay, and various chapters in your biology. I'll come with another video to clear out some more 4 chapters or 4 more topics that are very important as per your 12th syllabus, and I hope that you will be staying with me to watch these and learn from this, okay. If you still possess any doubts, please feel free to ask. I am always here to answer it, okay. Thank you for listening. That's it.